This recording is going to cover the cell, diffusion, and osmosis. Parts of a cell. We have already learned that the cell is the smallest recognizable part of the body. It is the unit of structure and function of the human body. The cell membrane or plasma membrane is the outer lining of the cell. It separates the cell's internal structure from the external environment. It is selectively permeable or semi-permeable, meaning that it permits certain substances to enter and exit the cell, which helps maintain the proper environment for the cell to function. There are two processes that we will study that will control the passage of materials through the cell membrane. They are diffusion and osmosis. Principles of diffusion. Before we can learn how substances diffuse across the membrane, we need to understand a little bit about solutions. Solution is a homogeneous mixture of two or more substances. The term homogeneous or homogeneous means uniformly mixed and distributed in the solution. There are two parts to a solution. The solvent. This is the part of a solution present in the greater amount, the part that does the dissolving. Solute is that part of a solution that is present in the lesser amount and the part to be dissolved. Think of a cup of coffee and sugar. The coffee is the solvent and the teaspoon of sugar you add is the solute. The coffee is present in the greater amount and it is dissolving the sugar. The sugar is present in the lesser amount and it is being dissolved by the coffee. So diffusion is the movement of molecules or other particles in a solution from an area of greater concentration to an area of lesser concentration until a uniform concentration is reached. For example, if you take a blue dye pellet and place it in a beaker of water, the pellet will diffuse or spread from the region of higher dye concentration to the region of lower dye concentration. This will continue until it is uniformly mixed and distributed. Notice that there was no mention of a membrane. Substances may also diffuse through a membrane if the membrane is passable to them. Osmosis. Osmosis is the movement of water or solvent through a semi-permeable membrane from an area of higher water or solvent concentration to an area of lower water or solvent concentration. Remember that water or solvent will move from a higher water concentration to an area of lower water concentration. Another way to look at osmosis is by looking at the solute concentration. So using this, osmosis is the movement of water or your solvent through a semi-permeable membrane from an area of lower solute concentration to an area of higher solute concentration. Osmosis can only occur when the membrane is permeable to water. If a red blood cell is to maintain its normal shape, which is biconcave, meaning indented on both sides, and the volume, it must be placed in an isotonic solution, which is a solution in which the total concentration of water molecules and solute molecules are the same on both sides of the semi-permeable membrane. Under normal circumstances, a 0.85% of NaCl or sodium chloride, salt, solution is isotonic for the red blood cell, thus allowing water molecules to enter and exit at the same rate, maintaining its normal shape. So let's see what happens to the shape of a red blood cell if it is placed in a hypotonic solution. This solution has a lower concentration of solutes and a higher concentration of solvent or water than the solution with which the cell is compared. Water molecules will enter the red cell faster than they can leave, causing the red blood cell to swell and burst. The process of rupturing a red blood cell is called hemolysis. It will be called plasmoptosis if you are referring to a bacterial cell. So now let's see what happens if we take that same red blood cell and we place it in a hypertonic solution. This solution has a higher concentration of solutes and a lower concentration of water or solvent than the solution with which the cell is compared. Water molecules will move out of the red blood cells faster than they can enter, which will cause the cell to shrink. Shrinkage of a red blood cell is called crenation. It is called plasmolysis when referring to a bacterial cell.